Before.
Hi everyone, I'm Natasha. I'm in Gulchen Pekranadz's lab. We do a lot of studies into neuronal metabolism, but today I'll talk to you about my specific thesis project, which is focused on metabolic plasticity and neuronal resilience, unraveling adaptive responses to chronic mtDNA damage. So our body relies on energy in the form of ATP. An organelle known as the mitochondria produces about 90% of this energy. Oh, sorry. And out of this pool of energy, the brain alone uses about 20%, which is crazy considering it's such a small organ. And out of that energy pool, neurons use 75 to 80% of that energy. So I want to introduce how essential mitochondria are for energy production in neurons. Neurons cannot store fuel, so they're constantly making ATP on demand. So a cortical neuron, which is just one type of neuron out of several different kinds, like hippocampal, dopaminergic, but in this example, cortical neurons alone use 4.7 billion ATP molecules per second. And at the presynapse, about 164,000 energy molecules can be used per presynaptic vesicle release. So over here, so in the neurons, mitochondria are present all throughout the cell. This is just one cell. And the mitochondria will modify their shape to meet these specific local energy demands. So here you have the cell body, this is just the axon, and this is the terminus of the cell. So the mitochondria are also unique in the fact that they have their own genome, which I'll refer to as mtDNA. And this genome is 16 kilobase pairs. It encodes 37 genes, 13 of which are essential polypeptides for 4 out of the 5 respiratory chain proteins. However, it's unknown if all the mitochondria in neurons contain mtDNA. And unfortunately, as we age, the mitochondrial genome is very prone to damage and mutations, which by extent result in mitochondrial damage. And the molecular changes that occur during the transition state from healthy to damaged mitochondria are not well understood. So I'm interested in the metabolic changes that trigger the progression of mitochondrial damage in neurons and how it contributes to age-related neurodegeneration. And if this is better understood, then we could develop therapeutics that work earlier before symptoms of neurodegeneration occur. So my first big question is, how does chronic mtDNA damage lead to neuronal decline? And from this question, I have a few other ones, such as what exactly is the distribution of mtDNA in neurons? And does altering mtDNA maintenance lead to mitochondrial dysfunction in neuronal processes? What are the functional and morphological consequences of aberrant mtDNA in neurons? And does metabolic compensation occur in neurons following mtDNA aberrations? I hypothesize that the distribution will be very heterogeneous throughout the different neuronal compartments. And when you affect mtDNA maintenance, it's going to lead to changes in the neuronal morphology, such as reduced dendritic branching, along with more fragmented mitochondria, as seen in other mitochondrial models. And to compensate for damage, neurons may upregulate amino acid or beta oxidation pathways to continue producing energy. So to probe mtDNA distribution in different neuronal compartments, I employed genetic labeling and immunofluorescence strategies. My first method was utilizing a protein known as transcription factor A mitochondrial, or TFAM for short, as a way to label mtDNA since it binds to mtDNA in both its active and inactive replication states. And another method I'm testing is the use of what's called an MT highness reporter. It has a mitochondrial targeting sequence attached to a photoconvertible fluorophore, and it's specific to mtDNA. So for the first tool, I first isolated neurons from E18 rat pups and cultured them for about 2 weeks before introducing the genetic tools. And so for the first tool I tested out, which was TFAM connected to an M-Clover fluorophore, I found that about 50% of axonal mitochondria contain mtDNA versus 80% in the dendrites. I was also curious to see if mtDNA was more present in stationary versus motile mitochondria and the axons. I was anticipating that it'd be more present in stationary mitochondria, but I found that it appeared to be present in both mitochondrial populations. So while this tool is great, the issue with it is that I'm overexpressing protein that's involved in mtDNA maintenance, but I'm not accounting for the endogenous population of TFAM in the cells. So that's where the other tool known as MT-Highness comes in. And we were generously gifted this tool by Dr. Timothy Schutt. And basically how this tool works is it has a preference for AT-rich sequences. And despite the bacterial history of mitochondria, the core machinery is actually very viral instead. It actually is derived from T phages. And so the Highness reporter is going to be able to bind to mtDNA because it prefers AT-rich sequences, but it should not impact any of the replication or transcription machinery. So I tested this tool out in COS cells, and I showed that not only does it seem to be present in every single little mitochondria in COS cells, but I can also use its photoconvertible ability. So my next step is to continue testing this in neurons, see if it's a good tool to use in neurons, because neurons can be very tricky to work with. And lastly, for this section, I wanted to validate the results of labeling mtDNA with a DNA antibody. The Pelot lab showed that this antibody can be used in neurons. However, their labeling was done in very young neurons and in high glucose conditions, so not physiologically relevant, which is an issue with the whole neurobiology field. So I used this antibody in my own cultures and saw that it does seem to localize to the mitochondria. The only issue with this staining is I didn't stain the whole mitochondrial network. I only did low efficiency labeling. So my next step is to label the entire mitochondrial network in neurons, confirm that this DNA antibody works. All right, but studies have shown that with an increase in age, there's a reduction in mitochondrial DNA copy number in aged versus young whole brain tissue. And it's even more prevalent in the case of neurodegenerative diseases like Alzheimer's disease. And it's also been observed mtDNA maintenance proteins are also impacted. So I wanted to develop a model to kind of recapitulate this. So I had a couple strategies. One is overexpressing a dominant negative polymerase gamma mutation in mature neurons to induce mtDNA depletions and deletions, or conditionally knocking out TFAM in neurons isolated from these TFAM flox mice. But for the purposes of today's talk, I'm just going to focus on the first strategy. So polymerase gamma is the only mitochondrial DNA polymerase, or at least the most well-studied. And not only does it have a function in replicating mitochondrial DNA, but it also has an exonuclease function. So it's the main repair mechanism if there are mutations in mtDNA. So in my model, I said that I'm introducing a dominant negative mutation. I'm actually going to be focusing on these two specific residues in polymerase gamma. These two residues are carboxylate ligands that bind to magnesium. They're essential for nucleotide incorporation. So D890 helps the metal associated with a nucleotide enter the active site, and D1135A is involved in the finger closing transition. And it's been shown that mutating these regions can impair bacterial growth and promote tumorigenesis and neurogastrointestinal disorders. And another lab, the Spellbrinks lab, actually tested the effects of mutating these residues in HEC293 T cells, and they did see what they were expecting, a reduction in mtDNA copy numbers. But they have this amazing tool, but they never actually visualized polymerase gamma in the cells. So I used their tool and I conducted some immunofluorescence and saw that I can express both the wild type and the mutant ones. I did a quantification, and basically I'm just confirming that this polymerase gamma protein co-localized to just the mitochondria, even after just 48 hours of expression. The next step was to determine if the mutations would even express in neurons, because like I mentioned, they can be a little tricky. So I allowed the neurons to express these mutations for 5 days in culture before staining for polymerase gamma, and found that this plasmid does express in neurons, and you can see that they are localized to just the mitochondria in the more punctate form that I expected. So I do have some preliminary analysis on the mtDNA content in neurons expressing these dominant negative mutations using the TFAM mClover tool. And I saw that in dendrites specifically, there appears to be more clumped nucleoids. So I measured the area of TFAM per dendrite area and found that the TFAM area is higher in this particular mutant, which is the D1135A mutant, compared to wild type. However, this is not what I was expecting to see, and I believe that this could actually be due to the fact that I'm overexpressing another mtDNA maintenance protein, which could be acting as a rescue in these mutant conditions, hence why I'm seeing an increase. So this is where those other tools would be super valuable. So next, I wanted to observe if there were any changes in neuronal structure, especially the dendrites, because we know that with mitochondrial insults, it ultimately results in altered mitochondrial distribution, which can lead to dendritic degeneration and eventually neuronal degeneration. So for this, I looked at my cells and noticed that there was something different in the dendrite morphology in my two mutant conditions versus the wild type. So I used Imaris to do a 3D reconstruction of each cell and compared the branching through shoal analysis. And from these reconstructions, I saw that about 100 to 200 microns away from the center of the cell, the neurons have much more reduced branching. Another phenotype I also noticed was a change in dendritic spine density, specifically in just this one mutation, the D890N pol gene mutation. And so I compared the number of spines, just quantified it, and I saw that, yeah, it does seem that there is an increase in spine density for specifically just this mutation. I'm going to actually use Imaris to further quantify and classify the types of spines present in each mutation model. So while the D1135A mutant model didn't seem to have didn't have much of a dendritic phenotype, I did notice an axonal phenotype. So further, like distally located from the cell body, I noticed these swellings, and there appeared to be an accumulation of mitochondria and some pol G signal. And this was really interesting because this data is actually recapitulated in dopaminergic neurons of a Parkinson's mouse model another graduate student in our lab is working on. So after investigating the structure, I next wanted to know if neuronal function was affected by these polymerase gamma mutations. And so we know that with neuronal functions such as vesicle release, it requires on-demand ATP synthesis. So in our lab, we can measure on-demand ATP synthesis using a tool known as IATP sniffer, whose fluorescent intensity changes in response to ATP concentration. So with IATP sniffer, we can see how well neurons adjust to their energy production by just stimulating them to induce activity. So neurons after stimulation will then compensate for energy loss by increasing ATP production so that levels gradually return back to baseline. So this drop in recovery is really ideal because we can assess whether neurons are capable of sustaining on-demand ATP synthesis. And so with the wild type, which is here in gray, I do see a drop, a recovery. We stemmed it twice, so we see another drop and recovery before we added metabolic inhibitors, which should completely shut down ATP synthesis. However, in our two mutant models, we saw that even after this first stimulation, that ATP synthesis levels just never recovered. So this was very interesting. It shows that there is something wrong with mitochondrial function. So my next step is actually to look at mitochondrial DNA, how mitochondrial DNA defects impair mitochondrial function. A couple of ways I'm planning to do that is through a seahorse assay, which measures oxygen consumption rate in the cells. So if you remember in oxidative phosphorylation, oxygen is the final electron acceptor. So without oxygen consumption, you're not going to get any ATP synthesis. So if it is impacted, I expect to see quite the reduction in oxygen consumption levels. Here's just an example from one of our papers. In this case, we saw the opposite, where if we kind of basically increased glycolytic activity, we saw an increase in maximal respiration of these cells. And usually when you get this data, you'll see this curve. So we add a bunch of different drugs to inhibit the neurons or the cells at different time points. And each of these drugs do different things. So oligomycin should shut down ATP synthesis, but FCCP allows, it basically creates holes in the membranes of mitochondria to allow protons to flow through. And that's how we get like what is called maximal respiration of the cells before we completely shut down the entire electron transport chain with antimycin A and rotenone. Another assay I'm planning to do is actually investigating mitochondrial health. One way to do that is to look at mitochondrial membrane potential using TMRM staining. Basically, what I anticipate to see is very reduced mitochondrial membrane potential. And lastly, we know from studies with other polymerase gamma mouse models that metabolic rewiring does occur after induction of these polymerase gamma mutations. However, I want to investigate the changes that are occurring specifically at the neuronal level, as most proteomics and metabolomics are done in full tissue samples, and sometimes they don't even focus on the brain. So eventually, I want to see what metabolic pathways are up or down regulated via metabolomics and confirm the exact pathways through proteomics. But with that, I showed that mtDNA molecules are heterogeneously distributed throughout neurons and are present in both stationary and motile mitochondria. The mutations to polymerase gamma result in less dendritic branching, and overexpression of a D89DN polymerase gamma results in increased dendritic spine density, and overexpression of D1135A polymerase gamma mutation resulted in these swellings in the distal axons, and both of these mutations affected the neuron's ability to sustain on-demand ATP synthesis. But with that, I'd like to thank my lab. I'd like to thank Dr. Pekkernas for her guidance and mentorship. And I'd like to thank, once again, Dr. Schutt for providing the MitoHinus tool. I'd like to thank my committee members for their guidance. And I was funded for a couple of years with the T32 training grant, and I still like to give thanks for this grant funding my project. And that, I am open to any questions you guys might have.
For the IATP sniffer, biosensor, how is it detecting like specifically ATP? Like, is it just like a pH thing, or is it like binding to ATP, or like?
It's binding to the protein, ATP synthase. Basically, it's yeah, it is binding to ATP as a result, basically.
I was just curious.
No, it's okay. It's a really cool tool, actually.
So there are like other like breath sensors too,
Yeah,
but I don't think that those are super
yeah.
good.
No, the IATP sniffer is definitely one of the more popular tools. They've actually come out with like a mitochondria specific version. So like the tool we have, you saw, was like basically very cytoplasmic. They came out with a tool that's very like mitochondria focused, which is super cool. Any other questions?
Sorry, follow-up question on that. Has there been any studies to show like how specific it is for ATP as opposed to ADP or AMP?
What? Oh, for the IATP sniffer
Yeah.
tool, it should bind to just specifically ATP.
So, in neurons, the mitochondria is also located in the axon, right? Like, that was your point. Would you expect, this is very speculative, but would you expect for myelinated neurons that it wouldn't exist in the myelinated region because there's just
No, it should always be, mitochondria should always be in the axons. It's how it's fueling energy and synaptic vesicle release because axon's huge and it's forming all of these different synaptic sites. So you got dendrite binding to like, not binding, sorry, a dendrite kind of like communicating with an axon at some random region. And they'll be like, basically transmission occurring there. So yes, there should be because you need mitochondria to fuel all of these different processes.
No, that makes sense, but I'm curious, like locally by the myelinated part, if they're like, you would expect some of the localization to be in these like nodes of Ranvier or like closer to the nodes.
I'll admit, I'm not super familiar with
Okay.
like myelin sheath biology. That's definitely really interesting. I know Wendy Campana's lab is working on that.
Yeah, no, sorry, just a very speculative question.
No, it's fine. I appreciate it.
Question, so this basically should also affect the action potential of the steroids.
Mhm.
Do you think? Is the effect in the morphology that we observe in the axons have more effect on that action potential? I mean, have you thought about studying this or have you guys studied like being a patch clamp and just or looking at ion channels or like, some calcium flux, like
Okay, actually, calcium flux, we do have a tool for that, GCAM. That's something probably I will do. In terms of patch clamping, our lab is not equipped for that.
our lab has it and talk to Dr. Jog, you have a collaborator. They're all like patch clamping and like electrode recording, so. Or this in different conditions.
Okay, yeah, no, that'd be great. I'll talk to my PI about that.
I was curious, you said your lab also studies like a Parkinson's mouse model.
Mhm.
I'm not very familiar with the polymerase gamma, but do you think, like, do you see polymerase gamma dysfunction in that model, or do you think that, like, there's a different mechanism?
I think that's a different mechanism specifically. So in Parkinson's model, usually it's actually TFAM, that other protein that's more commonly affected. So in those models, you should see like a reduction of TFAM. That graduate student, yeah, he hasn't focused on polymerase gamma at all, but he has actually been doing mitochondrial DNA staining in whole tissue and is actually noticing a very interesting phenotype in specifically the swellings that he's observed. But that's all I say about that.
Thank you, Natasha.
One.
Okay. Hi, everyone. Sorry for some of those technical difficulties. My name is Dylan. I am a second year student in the interfaces program. I'm a third year PhD student in mechanical engineering. And I'm excited to talk to you today about my research. The title of this talk is Optimal Dosing Regimens from Multiscale PK/PD Models Using Safe Control and Safe Reinforcement Learning. So before I get started, just out of curiosity, during undergrad, did anyone take like a signals and systems class or systems and control class? Okay, cool. So when I was in undergrad, I wasn't in mechanical engineering, I was in biomedical engineering, but I took a systems and controls class. It was like required for us and I really liked it. But I had pretty much no clue why it was useful for medicine. And I hope by the end of today, I can convince you that I think control theory is going to be important for the future of medicine, particularly in pharmacology. So I'm just going to start by saying a lot of thank yous. So I want to thank my advisors in MAE. These are Dr. Sylvia Herbert and Dr. Boris Kramer. And then I also want to thank my interfaces domain science advisor, Dr. Jean Zhang in pharmacology. So each of these professors is an expert in a field that's going to really contribute to this project in a very fundamental way. And I'll try to point out where each of their expertise comes in. But I also, just before starting, want to give thanks to my fellow PhD students. And I'm going to shout out Will Sharpless and Nate Linden here, who are fellow interfaces trainees. They're former interfaces trainees. And you'll see some of their work pop up in this presentation as well. And then I want to thank the interfaces co-directors, the program coordinator, Kelly, and the interfaces program at large. So you're going to see this is a very, very interdisciplinary research area. And this just wouldn't be possible without programs that really prioritize interdisciplinary research. So even for those who haven't heard of control theory, you've probably heard the term systems biology tossed around. And these days we think of systems biology a little more in the context of like analyzing omics data. But actually at its inception, systems biology came out of the control theory community. So at least according to Wikipedia, this was the first, this conference was the first time that the word systems biology appeared. And this is kind of a cool passage from this conference proceedings. They talk about Norbert Wiener, who's one of the most famous control theorists, who wrote a book, Cybernetics on Control and Communication in the Animal and the Machine. This was way back in 1949. And in this book, they also talk about, or sorry, in the proceedings, they also talk about the use of systems biology in ordinary differential equation models of neural networks, which my understanding is a lot of where the research on artificial neural networks grew out of. So kind of cool to see where this field has come. I think later on, especially a lot of physicists got involved in this. And so if you've ever taken like a systems biology class, you might have seen this textbook. This is the kind of systems biology I'm going to be talking about today. So not neural networks, but transcriptional networks within cells. And basically, if you open up this book, you'll see a lot of diagrams and equations like this. So here, this is representing some transcription factor X, which regulates production of a downstream protein Y. And the idea is I can mathematically model the concentration of Y in my cell according to the concentration of X via this simple ordinary differential equation model. Many of you are probably wondering what happened to mRNA. You can ignore the mRNA in this model. It has to do with timescale separation. I'm going to get to that later. But once you have an ordinary differential equation to model a single gene, you can start putting these genes together. So here I have something called the toggle switch, which is this 2-gene module, the repressolator, which is this 3-gene module on the right, and then this thing called an incoherent feed-forward network on the left. And each of these has specific biological functions. And then once you start wiring these together, you can make complex logical operations. So this is kind of like the regulatory logic that works in your cells. And we have pretty good ordinary differential equation models to describe how these regulatory networks work. Okay, and today this is really important in the study of quantitative systems pharmacology, which is basically looking at these models for pharmacological applications. So QSP really consists of 2 parts. There's pharmacokinetics. This is what the body does to a drug. So think about things like metabolism, absorption of the drug, elimination from the body. And then there's pharmacodynamics, which is what the drug does to the body. So this is actually like how the drug makes an impact on a cell, for example. So this is typically by binding to a protein to change its activity. And then often these are transcription factors that they're binding to, which changes the downstream regulatory logic of the cell. And together, when you put the ordinary differential equation models that model your PK and PD parts separately, and you smush them together, you get what are called PK-PD models in the QSP field. And so this sounds very academic. I'm talking about ordinary differential equations models for biology. This seems like something that might be done in a corner in a math department. But the reality is this is really important in industry these days. So if you want to be the director of QSP at GSK, you might want to get your PhD in applied math or engineering. And you might want to get a proficiency in mathematical modeling, especially with ODEs and parameter estimation. If you want to stick around in La Jolla, you can do the same thing at Pfizer or at Merck, or you can go where one of my former colleagues from an old lab went, Takeda, after they graduated with their PhD, working on their QSP team there. And so a QSP is really important from a scientific point of view, but also for these companies, it's really important from a regulatory point of view, because the FDA is paying more and more attention to these modeling and simulation efforts when approving various like drugs and compounds and things. Okay, cool. So where does control theory fit into this whole framework? Well, let me just talk about what control theory is for a second. So we're going to take our biology hats off and put our engineering hats on. So if you've ever turned on cruise control in your car, there's an algorithm that is basically telling your car how much brake or throttle to apply to maintain a desired speed. If you've ever taken a commercial flight, you can think a control theory algorithm for getting you to your destination. So between takeoff and landing, control theory is running the plane. The only thing the pilots are doing in most flights is the takeoff and landing part. And we colloquially call that algorithm the autopilot. Or if you've taken chemical engineering classes or worked as a chemical engineer, you know how important process control is. And these are kind of like the traditional applications of control theory. But these days, we're asking a lot more from our control algorithms. And this is in part because we can ask a lot more. The rise in computing has allowed us to do many more complex things. And also the rise in deep learning has allowed us to use more sophisticated learning techniques, such as reinforcement learning. So if any of you have seen the Netflix documentary on how the Google DeepMind team built an AI algorithm that beat the world champion at Go, this is a control theory algorithm called reinforcement learning. And everything I'm going to talk to you all about today is deeply related to reinforcement learning and has kind of like a reinforcement learning flavor. So if anyone's interested in this type of learning, feel free to come up and talk to me afterwards. But in particular, now that we have RL, we can do many more advanced control things. So this includes self-driving and autonomous robots. And today I'm going to talk to you about one particular control theory algorithm. It's called Hamilton-Jacobi safety analysis. And this is really where Sylvia Herbert comes in. She did her training with the person who developed this algorithm at Berkeley. The first paper on this algorithm was written 20 years ago. And since that time, it's evolved into something like really just practically usable through really good software engineering. And today it's one of two main control theory algorithms used for safety critical systems. And so let me just talk to you about how this is used in engineering, and then it's going to become very clear, I think, in 1 second how it's used could be used in pharmacology. So let's say I have some mathematical model. This is an ordinary differential equation model for a drone. And I have some set of goal states. So I want my drone to land on this table, and I have some hazard states. So I don't want it to hit any of the objects during that process. So here, X is a vector which represents the state of my drone. So these are things like position, velocity of the drone. I just collect them all into this vector X. U is the set of all things I can do to affect my drone's motion. So this in particular is the power I supply to the motors. And D is all the disturbance. This is the thing that I don't know about the drone, anything in my environment that I can't predict a priori. So this might like be errors in your measurement of factors like the mass of the drone, or things like wind in the environment that make it hard to predict the drone's motion ahead of time. And what we're going to do is we're going to run this algorithm. And it solves, the way this algorithm works is by solving what's known as a Hamilton-Jacobi partial differential equation. And so here in this movie, G is a surface that represents information about your hazards. L is a surface that represents information about your target. And then V is this critical thing. This is the main object that we look for in control theory, in optimal control or reinforcement learning. This is called the value function. And the reason we care so much about the value function is as soon as you are able to compute it, you get the optimal motor control for free. So in this case, I just look at my value function and it tells me exactly how to supply the right amount of power to each motor in the drone in order to get it to reach its target safely despite the uncertainty in the model over time. Okay, so now shifting back to biology, what does a PK/PD model actually look like? So here's an example of a PK/PD model. This looks like a lot, but it's actually a pretty simple one. Here I have two drugs that I'm considering giving to a patient, so A and B, and there are three compartments in my model, so gut, blood, and tissue. And then there's one protein, and it comes in a phosphorylated and unphosphorylated form. So these first 5 equations are the PK part, the last 2 equations are the PD part. And if I write my state vector X as just being the concentration of the drug and the protein in either of its forms in all of the compartments, and U as being the rate of supply of either drug to the system, and D being the set of things I don't know, so the parameter uncertainties, I can again write this system just as X dot equals F of X UD, just like the drone model. So if I go back to this drone model now, instead I plug in a PK/PD model. Here, the goal states represent the therapeutic states. So these are like the levels of phosphorylated protein I'd like to achieve. The set G represents the toxic states. So these are the set of concentrations in each compartment I want to avoid. And then using the exact same math now gives me the optimal dosing regimen rather than the optimal motor controller. So this is the amount of each drug to supply at each time point in order to make sure that my patient gets to their desired concentration of phosphorylated protein while avoiding hitting the toxic doses in between in a robust way. So let me show you an example of how this can be useful. So this is a very simple, very, very simple PK/PD model. Usually for something so small, you don't even need to use control theory. You can kind of guess the right thing. Here's an edge case where it's not obvious. So this, in this model, I actually have a tissue compartment that holds on to drug for longer than I'd like it to. And so there are kind of 2 obvious ways to approach dosing. The first would be a constant dose. So with the constant dose, I can achieve the therapeutic goal, but I can't do it without the patient experiencing toxicity in the tissue compartment. And this is because the tissue compartment holds on to the drug for too long. So the other thing you could think to do is to apply a very aggressive dose. So I'm only going to apply dosing at the maximal rate I can for about 2 days here. And you again get to the therapeutic dose, but you just hit the toxic dose at the very end. So you're doing a little better, but not quite there. Now, if you apply Hamilton-Jacobi safety analysis to this, what the computer tells you to do is actually to use aggressive dosing for about the first 2 days. This allows you to build up protein. And then in order to make sure that the tissue compartment has time to metabolize the drug in the tissue, you lay off for 4 days, and then you return 2 days later. And this way you can get to the therapeutic goal without ever crossing the toxic threshold. So this is an example of how optimal control can be useful. As these models get more complicated, especially in the context of combination therapies, these become more and more essential. And this is the type of thing that they're trying to do, right? Get these like the right combination therapies, right timing, all these things in these QSP departments in industry. And so this, when I show this example, it makes it seem like it's more useful in the clinic, but I would actually argue that this kind of algorithm is most useful in the research phase when you're trying to evaluate like the highest impact combination therapies to apply. And like basically in your model, before you even have a drug, you might have a target you want to hit, and you might want to see in your model, is it actually a good idea to try to be pursuing these targets or this combination therapy or such before you actually begin any experiments. Okay, so I just told you that these companies are investing huge amounts of money in building really good models of PKPD. And I told you now that we have an algorithm that once you have a model gives you the optimal dosing. So what is there actually left to do? Turns out a lot of things. When you go from an algorithm that was designed for robots to an algorithm that was designed that is being used for biological models, there are several issues. The first is that this algorithm is really slow and inaccurate for big models. So if you're doing these computations explicitly, you can only get to about 6 to 7 dimensional models. To go higher, you need to start using learning algorithms, which are less accurate. And this is a big issue in biology because the models are typically quite huge. So we came up with 2 kind of essentially like model pre-processing steps that allow us to handle biological models. So the first is a singular perturbation approach. So this is basically a way of focusing on one time scale in your model. So if you think about biological models, they often are multi-scale in nature. They have things happening at the millisecond range all the way up to hour ranges. So here we're basically just going to focus on the time scale you care using this algorithm. And this paper was recently accepted to this controls conference, which we're very happy about. We're also working with Boris Kramer, who's the model reduction expert in MAE, on a way to summarize molecular level information into pathway level information. So that's something in progress that I'll talk about. And then this other kind of issue is that HASA is not yet developed for complex tasks. So if you remember the example I gave you before, I was just getting up to my therapeutic threshold, which is kind of what the algorithm is designed to do. But in pharmacology, I don't want to get to a therapeutic threshold. I want to get there and stay there in a safe manner. So we've developed this algorithm further so that it can handle these complex tasks that maybe don't come up as much in robotics. And so this led to this task decomposition algorithm, which is submitted to the International Conference on Learning Representations. This is one of like the big machine learning conferences. We're crossing our fingers that it gets in. We don't know yet. So this is what I've worked on so far. This has been this past year of this project. Starting the coming months for the next year of the project, I'm going to be applying this method to a real world PKPD model. And this is going to be specifically for KRAS G12 inhibition in non-small cell lung cancer. Why are we working on this model? Because Jin Zhang, who's our collaborator in the pharmacology department, is an expert on RAS signaling and also has biosensors for detecting RAS activities. So we can actually use their data to parameterize these PKPD models. So really excited about that. With the rest of my time, I'm going to talk to you about, at a very high level, about these 3 algorithms we built in part 1. So the first thing, as I mentioned, is that PKPD models are usually multi-scale. So they'll go from everything involving protein binding and unbinding up to translation and degradation of proteins, which operates on the same equilibrium cycle as the cell cycle. And so this is quite a big issue if you're trying to do computations across these scales. And so what we do instead is we use this timescale averaging that essentially averages over the fast scales and gives that summary information to the longer timescales. So here's a pretty simple example. I have a transcription factor T, which can be either in the phosphorylated or unphosphorylated forms. It's auto-repressive, so T star, the phosphorylated T, can bind to the promoter for the gene that encodes T, preventing further transcription of that gene. And the idea here is that if you are working at a set of parameter ranges where these processes, so transcription, translation, and phosphorylation are on the same timescale, which they usually aren't in real systems, then this is inherently a 2-dimensional model. So these are showing the contours of your value function. But you can mathematically prove that in the situation where these timescales are well separated, your value function becomes essentially one-dimensional instead. So I can wildly more quickly compute this value function. Now, this isn't such a big deal going from 2 to 1, not such a worry. Here's a bigger example. So this is a metabolic network example where I have a huge number of metabolites in this pathway, from some upstream metabolite M1 to a downstream metabolite P. And P is incorporated both into biomass in your cell and into the production of some transcription factor X I care about. And if I'm using explicit methods, which is what I'm doing here, the most powerful supercomputer in the world can't calculate the value function for this kind of thing in the amount of time that the world's been around. But now I can calculate it on my laptop because I'm able to reduce this system from a 25-dimensional system to a 3-dimensional system. And so what you ultimately get is this value function here. And what I'm showing here is the yellow dots are an inner approximation to the set of states where I can reach my target, which is this gray square. And the purple line is the outer approximation. So if I start within the inner approximation, I know I can reach my target. And if I start outside, I know I can't, and this is reflected here. So basically the idea is I've been able to effectively compute an approximation for the full high dimensional value function with just this, this value function in 3 dimensions that can be computed really quickly. So this is the kind of thing that we're trying to build up, methodology we're trying to build up to be able to analyze these PK/PD models. The second part we worked on is this compositional value function approach. So the idea here is in pharmacology, as I mentioned, usually you don't just want to reach some desired transient dose, but be able to maintain some sort of long-term dose or long-term concentration of some protein. And the way this works is by putting these value functions together in a kind of complex manner. And just for the sake of time, I'm just going to show you. If we return back to like a similar model to that model before, now I can not only get up to my therapeutic dose, but ensure that I can stay there without ever hitting my toxic dose, which is kind of a more realistic application case. And you might say, isn't this just what you would expect? You do the first thing first and the second thing next. And the reality is that's not the case. You have to work backwards using the value function from the second part of your analysis in order to inform the first part. And then finally, I'm going to tell you about this model reduction algorithm. So this is going from molecular information to pathway level information. So some of you may recognize this model. This is a model developed by Nate Linden, who's a former interfaces trainee that he did some pretty sophisticated computational analyses on. And the cool thing is this model, we were able to, it's small enough that I can actually compute the true value function for it. But what we can show is we can approximate it with an even smaller dimensional value function, essentially summarizing the map K dynamics into 1 or 2 states rather than the 3 states it normally lives on. And if people are interested in this, I have more examples where this works really poorly. And so we're trying to fix that, which is what's left on the methodology part. But for most things, it works pretty well. So next steps are finishing up this model reduction algorithm, and then second, applying all this work with the context of Jean Zhang's data. And so again, I'm just going to thank everyone for their support on this project. I mean, really, I've had so many people contributing expertise to me to be able to make this all happen, and the relevant publications are here. Yeah, happy to take any questions now. Yeah.
For the work with Jean, is like the specific readouts that you're using to like inform the control theory model that you have, like the mathematical model, is that like are you using like data from like the actual like sensor that she has for this, or and like you're basically like looking at the time points, like when she treats the cells with the drug versus not, like how are you using that to like inform?
Yeah, I think that's all going to be decided in the coming months. So we're going to figure out what data they've already collected from their RAS biosensors and how we can use that for getting parameter estimates. But the basic idea is that they can get kinetic data with these biosensors. So they have essentially like fluorescence time courses and things where you can see the biosensors decaying in activity over time or rising in activity over time. And those kinetics are the things that inform these parameters in the model. So we're going to go from fitting essentially those curves to trying to get estimates here. And then those disturbance values, the parametric uncertainty is going to be related to like the standard error about those estimates that they have.
And then or like, I did a rotation in her lab, so I'm just like kind
Yeah.
of curious about. Some of it, so like, it's like, from what I understand, most of this stuff is just like on a single cell level there, and so like she doesn't work with tissues or. Like, animal models or anything, so like, how were you translating that into something like this, where you're looking at blood and tissue?
That's such a great question. This is going to be mostly on the PD side.
Okay.
It turns out that that's actually typically where you need most of the data because companies are forced to publish PK data.
Mhm.
So these drugs are already in development. Hopefully, I'm hoping I can find the PK data already available for them, but we need to get the PD data. You're not required to release PD data.
This is not kind of tangentially related, but I was wondering, I'm assuming, you have to change drug amounts and dosing based on like age groups and maybe gender.
Yeah.
And as we're moving towards an era of more like personalized medicine, is there some kind of way we can translate this to a personalized method where we change dosing based on whatever specifics?
Yeah, totally. That's a great question. So if you are able to make measurements or get solid estimates for parameters in your model, then you can just on the fly adjust the model, right? Like the idea is to be able to do this quickly enough that for an individual patient, if you can get a really good estimate of some critical parameter here, you can just reevaluate their optimal time course. And in fact, not only do that at the initial time when they're planning their whole dose, but in follow-up. So if the patient isn't responding as you expect, being able to adjust on the fly their dosing as the time course continues. That said, typically it's really hard to figure out what the parameters that are important are. So another thing that these analyses can be used for, if you can handle the dimensionality issues, is that they'll tell you what the most important parameters you have to identify are. So this is where, like in the research phase, using this kind of analysis tells you, hey, I really, really care about K here. Gamma 3 just basically doesn't matter at all. So this is kind of like doing a more sophisticated sensitivity analysis where you're planning for sensitivity with knowing that you're going to have to do downstream things later on. Yeah, that's a great question.
How much more computationally expensive would it be if you were to also take into account off-target effects, where you would have to look at on the PD side, where you're looking at like also the binding of your drug to another protein?
Way harder. Yeah, so every dimension that you include basically exponentially increases the computation time if you're using explicit methods. So these explicit methods basically are solving a PDE. So every dimension is another axis in your PDE grid. The cool thing about reinforcement learning, I mentioned this is all related to reinforcement learning. Reinforcement learning is a grid-free approach to this. So you can do RL in as many dimensions as you want and try to get that same value function through a different computational approach. But RL has these big issues with generalizability, right? So as soon as you move from the simulation to the patient, you might be sampling the patient might be a sample in state space that the RL algorithm has never really explored. And for safety critical things, we'd prefer to avoid those kind of issues, which is, yeah, where this balance kind of comes from. Cool. Thank you all so much.